Hello, this is a video on the structure of DNA power focus area. First, we'll be talking about what DNA is. DNA is the building block for how physical traits look and things that will make you, you. They are put into your genes and are what will make parts of you, right? They set you as you. DNA is what is known as a nucleic acid. These are molecules found within the nuclei of cells. They generally dictate, these nucleic acids will dictate how the cell works and what they'll do. And there are two primary versions of them that you'll learn about through high school. You'll learn about DNA and RNA. Now, we'll be primarily focusing on DNA for this topic, but RNA, it's generally similar. Now, one of the cool things about DNA that we'll do is it goes through a process called replication. This is what it sounds like. The DNA will duplicate itself or replicate itself. It follows a couple main steps. First, it breaks the bonds between the two bases. And so these are the two strands that you see following along the lines there. Right here. It will break these apart. Next, it will take a polymerase, and don't worry about that for now, but you know is what its main feature is. When it's with this two separate strands, this thing will then add complementary bases to the two sides. So they'll just basically reconnect them on as two separate strands. Now these two separate strands actually go through slightly different processes when they're replicating themselves. One is continuously linked, and we'll go through the entire process on its own real quick, and this is known as the leading strand, while the other part, will lag behind and connect itself in segments with gaps between each segment. This is known as the lagging strand. Now, the segments between the lagging strand will reconnect itself with an enzyme called a DNA ligase. Now, we'll start talking about more about what is in these strands in the next slide. Structure of DNA. This is the structure of DNA. Now, with DNA, it has a few main things you need to remember. It is a nucleic acid, as I said on the previous slide, and these nucleic acids are generally made up of something known as a nucleotide. Now, with the nucleotide, it has a few main components you need to think about. Here, so a 5-carbon sugar, a phosphate group, and a nitrogen-containing base. Here's a little diagram of what these are right here. This is what a nucleotide is, essentially. Now we're going to start talking about these nitrogen-containing bases, because this is where some real interesting stuff goes on. As you can see, structure here on the side, and this is where we're going to start talking about these bonds right here. This is what these nitrogen-containing bases do. This is what these are right here. These have, there's 4. They follow something known. There is adenine, cytosine, guanine, thymine, and these are all kind of shortened down to A, T, C, and G. Okay. They follow this rule that essentially explains how this bonding goes on right here, and it's pretty simple. A here will always pair with T, as you can see right here, and vice versa, of course, while cytosine and guanine will always pair together. So it's adenine, thymine, AT, cytosine, guanine, CG. Right? As you can see right here, follow along this base. That is the structure of DNA. Next, we will be covering things like amino acids, proteins, and then genes and traits, as well as going over a couple questions within the power focus area itself. Here we're going to talk about the amino acids and then cover proteins because these are linked together quite well. First, amino acids. These are made up of two main properties: an amino group and a carboxyl group. The sequences are basically limitless. You can kind of see a lot of the types of amino acids that will make up a sequence over here in this little image. Amino acids will usually be what makes up a protein. The two main parts of an amino acid being the amino group and the carboxyl group are covalently bonded to a carbon atom, with that carbon atom being bonded to a hydrogen atom and an R group. Now, an R group is kind of what sets the amino acid apart. It kind of gives it its own set of chemical properties. It kind of gives it its own shape. You'll probably see it referred to sometimes as a side chain. It's just another name for it. Now, we're going to go to proteins here. What is a protein first? Well, a protein is a group of amino acids made up of about 100 or more amino acids. Proteins are what does the thing the DNA is saying to do, basically. Your DNA will be like, your eye color is blue, let's say, right? And proteins are what does that, makes your eye color blue. Now, a protein can be cut into four main parts, okay? First, the amino acid sequence. This is what makes each protein unique and kind of defines what the protein does. So, hair, eye, whatever, right? The next part, the substructure. This is right here, as you can see in the little nice image we have here. This is the shape of the, basically the shape of the amino acid sequence from the primary one here. Kind of decides how it's shaped, essentially. And these are usually tied with like the hydrogen bonding from the amino acids, right? This is usually kind of tied together, kind of that bonding will kind of help decide the substructure, essentially. Next we have the tertiary structure. This is the overall shape of the protein and is made up of the secondary structure. And so you get to see as we go down the lowest here, the primary one makes up the second one, the second one makes up the third one, right? So that's kind of how it goes over. Basically, this third structure is just made up of the second one. There's like a bunch of little groups together. And then finally we have the final part here. This is an interaction between the subunits of protein, specifically hemoglobins of blood. Subunits of protein are, back to primary structure here, are your chains. They're your amino acid chains. So yeah, that's what we have for your protein. Next we have traits here. Traits, pretty simple. First, how do traits work? Well, usually with two parents, right? They'll have, like I said, a code, DNA. Half of it will go, you take two parts of it, and then kind of go back, they go together. Again, a lot of how traits work link back to the DNA slides. So, just go back a little bit. We talk about DNA there. That's essentially how traits work. Now you have types of traits here. These are traits with usually more than one gene influencing them. These traits, by nature, are more complicated just because they're made up of other traits, right? There's not a whole lot else to say, right? They're more complicated because it's multiple different things working at a time. Instead of just one thing deciding it, a lot of the time you'll see this, the more common type. Next we have dominant and recessive traits. Well, dominant and recessive traits, it's in the name essentially. It can be a whole bunch of stuff, eye color, hair color, whatever. With dominant traits, so first we'll talk about dominant traits. Dominant traits will take effect essentially over your recessive traits. So if you have, let's say, blue eyes, brown eyes, right? If your blue eyes or brown eyes is usually the dominant one, so they'll take effect over blue eyes. So if one parent gives you the trait for brown eyes, you'll take that over blue eyes, right? Now we have recessive traits, and these are like only come together when you have only two recessive traits. So again, going back to this idea of eyes, you know, if both parents have blue eyes and give that to their kid, then the kid will now take the two blue eyes, right? The parent can have brown eyes and give a blue eyes trait. We can do hair color too. It basically, recessive traits will only come out when you see both of them essentially in your DNA, in your code, right? Both of these are usually defined with a very specific type of sequencing. So dominance will always have capital letters when defining them, while recessive will always have lowercase letters when defining them. So you'll see something like capital A, lowercase a, that means dominant trait, while lowercase a would mean recessive. Two capital A's would just mean dominant, and yeah, that's kind of how traits work. And then we go into the final part, the questions. We'll talk about that next. Okay, first here we have the first question. We just have to go back to the first slide. The DNA. We've already gone over it. The DNA is kind of what is the building block for a lot of this stuff. As we've gone over already, DNA kind of sets everything in the process. That's kind of what encodes everything. That's how you get everything. That's kind of what sets your traits. Here, what is the composition of a nucleotide? We've already gone over this. Nucleotide acid, nucleic acids. Pretty simple. First you have the phosphate group. Next you're gonna have the sugars, the 5 sugars. And finally you have the nitrogen base, the A, T, so on, right? It's all pretty simple. We've already gone over a lot of this. Which of the following processes does not occur during DNA replication? Well, we can start the first question here. Enzymes unzip the data and add nucleotides to the template strand. Well, we know they unzip, and we know that they do that. So, not that one. 2 DNA molecules are made up from 1 DNA molecule. Well, it's in the name, replication, duplication. You make 2 from 1. DNA molecule unwinds before the duplication begins. Goes back to the first step. It unwinds at first. We kind of already did that right over here. So, not that one. So DNA condenses into chromosomes. It's this one. DNA does not condense into chromosomes. Okay, what do the following pictures represent? It's the correct structure. Well, first we know it's not going to be B here. This only has 1. Remember, double helix. It's definitely not going to be C. True, that's 3 lines. Not double being 2. It's going to be A for double helix. Now, question 5 here. What do the following organisms contain DNA? Well, everything contains DNA. It's the building block for everything. Everything's going to contain DNA. There's really no exceptions on what organisms do not contain DNA. It's kind of what decides a lot of you, essentially. So everything contains DNA. Given the following sequence, TTT-AAA, which sequence is a complementary strand? Well, first remember, complementary things, it's always A to T, C to G, right? So that means it's already not going to be C or G. Those do not pair with T or A. I'm going to eliminate those right off the bat. Well, it's T to A and A to T, right? So we know it's definitely not going to be another set of T's and A's. So we can eliminate this bottom question, answer right off the bat, and we're left with A to T here. And yep, correct. A's to T's and T's to A's. And that's it. Let's just click here, finish, and there you go. Well, that's the diagnostic at least.